So yesterday I watched Spider-Man in the cinemas, and I wish I could have a pause button because I wanted to tell my children what exactly, the terminologies, the biological terminologies Peter Parker was saying, okay? So anyways, what happened was that, look, I'm telling you some of the basic terminologies, okay? All right. He is using two terminologies, actually three, okay, which is mRNA and then siRNA and then inhibitor of gene expression, stuff like that he's saying, okay. So basically in his body there's an enzyme arcanide that was produced, okay, and it was giving him headaches and all of that, and he was, he wanted to get rid of it. Anyways, so what exactly is the basic terminology he was using was that, look, over here this is the DNA strand, okay. Now DNA strand has the entire recipe, okay, in order to make the protein. Now, how exactly it works? It has two stages, okay. First of all, the first stage is transcription and other one is translation. Now, in transcription, as the name suggests, writing, okay, transcription is writing, right? Okay. So, let's say this is the DNA molecule, okay. Earlier, it was double helix structure like that. An enzyme helicase unwinded it, okay. Now, the DNA strand has been exposed, okay. And now, RNA polymerase will create an mRNA. Now, mRNA, as the name suggests, it's a messenger enzyme, okay? So now what Peter Parker was talking about was that, if he would block this mRNA, okay, then no more gene expression will happen, okay. What is the genes are the small sections of the DNA, okay. All right, so over here the genes, if the mRNA would be blocked, okay, by siRNA, okay. siRNA are the single-stranded DNA, single-stranded RNA, okay, that you produce in the lab, okay, and it can block the mRNA. All right, so mRNA would not be able to work. Now, how exactly this mRNA was supposed to work is this: it would go under good translation, okay, where ribosome would be deciphering what exactly mRNA has, okay. So for example, over here you see, if ribosome would decode it, okay, so three bases would code for one amino acid, yeah, okay. But let's say if this mRNA was blocked initially by siRNA, then this won't even like be produced by ribosome, like ribosome would not be attached, ribosome would not be able to attach on the mRNA if mRNA is already blocked, yeah. Okay, I hope you enjoyed the basic, the terminologies that I could break it for you.